Welcome to Wild Turkey Science, a podcast made possible by Turkeys for Tomorrow. I'm Dr. Marcus Lashley, Professor of Wildlife Ecology at the University of Florida.
And I'm Dr. Will Goolsby, Professor of Wildlife Ecology and Management at Auburn University. We're both lifelong hunters and devoted scientists who are passionate about hunting, managing, and researching wild turkeys.
In this podcast, we'll explore turkey research, speak to the experts in the field, and address the difficult questions related to wild turkey ecology and management.
Our goal
is to serve as your connection to wild turkey science.
Well, Marcus, this is what I'm excited about. I know that I say that about every episode because we talk about turkeys. I'm always excited to talk about turkeys, but it's long overdue.
How many times have we said we really should be talking about some disease stuff? But I mean, that has come up on like a dozen or more episodes at least.
Mm-hmm.
And I know it's one of the most common recurring questions that people have, just various aspects of disease ecology about turkeys and how much we should be worried about various things. And you and I have dodged those questions quite a few times trying to get an expert like the one we have today on.
Yeah. I mean, just, improve your habitat and turkeys will be more disease resistant, right? But we're so thankful to have Dr. Rick Gerhold here today with us that can do a much better job, do this topic, justice compared to us. And he is a professor of disease ecology at the University of Tennessee. And I know, personally, Rick has helped me out a lot with, consulting on various disease-related aspects of some of my ongoing turkey research. And he's been instrumental in helping further and advance my education in that arena. So, Rick, we're really glad to have you on today. Thanks for joining us.
Absolutely. It's my pleasure. So thank you for inviting me.
Yeah, Rick, I'm really excited to have you after being at the convention and watching your presentation. I immediately thought that you're the guy. We need to have you on to talk about various things you're working on, because you covered several things in that presentation, that are hot topics in the turkey world right now. So I'm really glad, that you were willing to do this and appreciate it.
Well, to me, it's my pleasure. So this is kind of stuff I really enjoy, getting to talk about science and getting information out to the public. So I really appreciate the invitation.
Yeah. So to start out, can you tell us a little bit about your background and how you got to where you are now?
Yeah, absolutely. So, I went out after following high school. I went to Purdue University. Not really knowing exactly what I wanted to do. Kind of went on general science, and one of my friends in the hall happened to be a wildlife biology major, and was always one of my passions. Growing up, hunting and fishing was what I wanted to do. Being outside, and still what I want to do. And so, finding the curriculum and looking at that, it was an instantaneous switch. So in the process of getting my degree, I had the fortune of having a couple teaching assistants that were doing various projects for their master's and PhD, and one of them happened to be working on the ecology of the raccoon roundworm, Balis ascaris. And so I was able to go out and help her trap raccoons, take samples, and became very fascinated about the ecology of parasites, and particularly in the interaction of parasite and host. And then got encouraged by a professor there to go on to vet school, which I did. I got my DVM from Purdue as well. And following that, I got very lucky that I just happened to be a job opening at the Southeastern Cooperative Wildlife Disease Study, which I'll refer to as SCWDS from now on, just because it's easier to talk about, as their staff veterinarian, where I did everything from general necropsies, which is what we call an autopsy on an animal, to capturing wild animals, experimental infections, training graduate students, is also coordinating the CWD testing for the Southeast for the first 7 years of the testing we did. And then just because I wasn't busy enough, I decided to go on and get a master's and PhD there. Both of them were in avian parasitology, particularly protozoa. And then following that, I got a postdoc working on Lyme disease here at University of Tennessee for 2 years. And again, being in the right position at the right time, as that was ending, a faculty position opened up at the vet school for wildlife disease parasitologist. And so I've been here in that position since 2013 and really enjoy it. A combination of teaching courses, mentoring graduate students, diagnostic services, and whatever other things my department head and dean can come up with. So, but I enjoy it.
Rick, I'm just curious. What is it about parasites that fascinated you? I know, but I'm aware of Balis ascaris, and it's a pretty wild life cycle. And I was thinking about it particularly. I think it's called a paratenic host. That's the intermediate.
Yeah, very good. Wow. Yeah. Maybe you don't need me.
No. I know one $3 word. That's it. So I was leading a project on raccoons with a guy in the vet school at Mississippi where we were just screening them for Balis ascaris. So I read up on it to try to learn it. I thought it was pretty wild, and there are various other really wild life cycles, I guess, for parasites. I was just curious, what about them got you excited about it?
Yeah, it's a great question. I think it has to be the ecology of the host and the parasite together. You think about how the host is trying to, you know, um, reduce the burden of parasites because obviously it doesn't want a parasite. And so it tries to have increased immune response to it. At the same time, the parasites trying to evolve to evade the immune system. And there's a term in ecology when it comes to parasitology that's called the Red Queen hypothesis, and that's borrowed from Alice in Wonderland, where the Red Queen tells Alice you have to run as fast as possible to stay in place. And so what's kind of the thought process is that is even though it looks like the parasite and host are kind of staying in this stationary, stagnant, they're both evolving as fast as possible, one trying to evade the immune system, one trying to stop the parasite. But together it looks like they have this kind of,
stable relationship.
Exactly. Perfect. Thank you. And so that fascinates me. I also am fascinated by things like brainworm in moose and elk, where the white-tailed deer, that's a natural host, has no disease. But then when the larva gets into the spinal cord brain of a aberrant or non-particular host, such as the moose and elk, it tunnels through the spinal cord, tunnels through the brain, eventually killing the animal, which in a way it belongs to that group of parasites called competitive exclusion parasites, where that parasite's actually helping its host, in that case, white-tailed deer, have more resources by killing off its competition, such as the moose and elk. And
I have studied up on that one a little bit as well because it's an important one for deer, which I do some work on deer. Don't they have to eat a snail to get
Yeah, they either have to eat the snail or slug, because the snail and slug are the required intermediate host, or they have to ingest something that has the particularly slug slime on it, in which it left the infective stage of the larva. So, but the other thing is we're actually doing a project right here in Tennessee with the Tennessee Wildlife Resource Agency, looking at our elk. I know we're talking about turkeys, but we're talking about our elk project, because we're not having as good a growth, and we have found that brainworm is the number one cause of infectious disease in them. Looking at gastropod populations, and I happen to we were very lucky here at Tennessee to have one of the world famous gastropod researchers, and he was telling me about many of the micro gastropods that you can't even see without um, a microscope that may be important, that an animal could eat many of those and you wouldn't even know it. And no one's even looked at those. We've always been looking at the macroscopic large gastropods.
So
So there's a
so
lot we
elk could
just,
literally be eating snails that are not visible to the eye,
yeah, yeah.
and
Most
get
of them are in the soil, um, but potentially as they are grazing, they can potentially pick it up. So there's a whole area of this field that I just have barely scratched the surface on. And then people are calling me like the expert for this disease. I'm like, I don't know anything about it.
you
So
know a few of the words.
yeah, that's
That's
all
about
I
all I
know
know.
the words, and I can lead you the right way, and I can help maybe diagnose it, but that's as much as I can do. But the ecology, there's still so much to be known about it.
Mm.
So that's kind of stuff that fascinates me about parasites, and then there's so many examples of that. Whirling disease is one of them. Uh, Interesting enough, blackhead histomonas is one of those, and then some of the other ones like Trichomonas gallinae in pigeons. All those have those weird things where the parasite can actually help the host by killing off its competition. So
That
Mm.
that's so weird.
yeah, it's wild. So that
I
coevolution of the parasite host is just fascinating, and the more you learn about it, the more you don't know. So
Yeah. Well, I certainly can appreciate what how fascinating it is, and how people get fascinated with it. Because the more I dig into various topics, it's I'm just blown away at how things work in those types of systems.
yeah, it's fascinating. I've actually had to warn undergraduates and vet students to come to work in my lab. I'm like, about half of the people that come to work in my lab change their major to parasites. I'm just going to warn you that. And I've had several of them like, no, I'm going to be equine vet, and one of them just got a job at Oklahoma State as their parasitology professor, and she actually did her summer research on turkey parasites, tapeworms, and got so fascinated with parasites that she ended up doing her PhD and now a professor. So it kind of sucks you in, no pun intended. So
I wonder if half of our audience will become parasitologists.
hey, we need
Or
we need
statistically,
we
one of
need
us
more.
may.
Yeah,
Yeah.
exactly. So,
Who's it
but
going to
actually,
be? It's going to be Will.
No.
I actually find a lot of the wildlife biologists who dabble in parasitology make the best parasitologists because they understand population dynamics so well
Mm.
that they can really think more on that broader scale than people who are used to thinking on an individual animal basis.
Mm.
So,
Hmm.
uh, quite a few, actually, I would say the three people who taught me the most about parasites were non-veterinarians who were either wildlife biologists or, went through as parasitologists, and they just really were able to think and got me to think about them a little differently and encourage me. So
Hmm. Yeah, that's an interesting way to look at it. Well, what about turkeys? What are the things that excite you about it, or maybe the emerging ideas or the emerging pathogens, whatever, however you want to to
yeah,
discuss it about turkeys.
yeah, turkeys. For something that we do so much research on, I really feel like we know we're just scratching the surface of understanding diseases in turkeys. There's certain diseases that I think are relatively easy to spot, such as avian pox. Um, Caused by a virus that's transmitted mainly by biting flies, but direct contact, turkey to turkey, can spread it. Or us that are not, if we're using the same equipment between two separate turkeys, one has pox, we can be the transmission source ourselves. Um, but those have those very characteristic outward, what we call lesions, having those gross abnormalities where you can see the bumps and the proliferations on them, sometimes so severe that it's impedes their ability to eat, to see. Um, And so that one is always something that's considered one of the very important diseases of turkeys. There's work. There's a couple other things that have been kind of getting more attention these days. One is common name is blackhead. That's caused by a protozoa named histomonas. Unfortunately, the blackhead is a misnomer because nothing turns black. I'm not quite sure where that name ever came from, but that's where the common name is. So if you're looking for a black head on blackhead, you're not going to find it. But it's got attention because of concerns of everything from use of spent chicken litter as fertilizer, which makes it very good fertilizer, and the concerns with certain types of chicken fertilizer. Not all of them are important sources of it. Some much more of a concern than others. And then in addition, other things like non-native species like ringneck pheasants can be a source of the parasite because pheasants are considered the natural host for this. They don't get sick from it.
For the blackhead.
Yeah, from the blackhead histomonas. But turkeys are exquisitely sensitive to it. And then there's another big field of study right now on this area of LPVD, lymphoproliferative viral disease, also REV, reticular endothelial virus disease. This is a real area that we really don't know much about because you can go and test wild turkeys throughout an area and you can get upwards of 90% of these birds positive for this virus by
Did you say
doing 90% positive.
Whoa.
But few, if any, have any disease symptoms, and few, if any, will ever have any disease symptoms as far as we know. Now, the ones that do have disease can have everything from lesions of their bones, lesions of some of their internal organs, whether or not it precludes them to be more susceptible to other infections. We don't know that. So this has been an area where one of my colleagues at the University of Georgia Wild Disease Study, Nicole Nemeth, has been really looking into trying to understand some of that, importance of it. As of right now, it's you can find it. You're definitely going to find it if you look for it. But how to interpret what it means has been the big question. No one really has a good handle on that.
So, Rick, what you're saying is a large proportion of a population might have it. They test positive for it.
They test,
and they may have, I don't remember the word you said, maybe symptoms of it, but they aren't expressed visually where we can observe it.
Most of them don't even have symptoms at all. They have nothing.
Oh, even internally they don't.
Not internally. Under the microscope, you see everything looks normal, but you can find the virus by doing molecular testing. You can actually detect it. What's called the proviral DNA. It's a PCR test that they can do, and you'll find it in a large proportion of birds. And you can dissect every last organ, look under the microscope, and everything looks completely normal. And so that's why we don't really understand what it means from the population health. And one of the biggest challenges has been we haven't been able to propagate the virus, to grow the virus in the lab. Certain viruses we can make what's called a cell culture, grow cells in a test tube or a flask, let the virus grow, and then we can do experimental infections. Unfortunately, we have not been able to get that to happen with this virus. And so that really has precluded us from doing the really needed experimental infections to ask the question, what happens when a bird gets this? And then what happens if a bird has this and you give it a secondary disease that generally doesn't cause much issue? Will that LPVD REV reduce its immune status that allows other? We have no idea. Yeah. So it's a lot of. I get asked this question all the time, and unfortunately, the answer we all give is we don't know. Yeah. And so hopefully, Nicole and their group can answer some of these questions. We can work together to answer a little bit more. Other areas is toxins. that, again, we don't know a lot about. How toxins may impact everything from aflatoxins that we worry about, especially with artificial feeding of animals. Depending on the time of year, summer, corn can grow aflatoxins really quickly, especially on, humid, hot times. And, there's concern of that having everything from direct health in animals, but also potentially reducing their reproductive potential, their egg laying, recruitment, the population. We're just starting our group to look at this because we're doing a general fertility study of turkey eggs right now, and we're trying to understand some of these issues. Then other things like neonicotinics. There's been some good work with mammals showing it definitely have some uh, effects on reproduction and some small songbirds that can be deadly to them. So it's the unfortunate thing with toxins. It's expensive to do the work, so you need quite a bit of funding
Hmm.
and two, without experimental infections of giving animals a certain dose in the lab and seeing what happens, it's hard to know exactly what a certain level means unless you can consistently see with this toxin level, I see this change in the liver under the microscope.
Hmm.
Uh, so it becomes a little tricky teasing out what exactly it means. So those are kind of some of the major players. Of course, there's other things, including, a few cases of polygranuloma, which happens from a bacterial disease. Mycoplasm, which is a bacterial disease. Concern of a couple other diseases, viruses, mainly from an aspect of transmission from domestic poultry to turkeys or vice versa is avian influenza virus and then Newcastle disease virus. So those are all kind of some of the players that we're trying to look at right now.
So, Rick, one of the questions that we get a lot is, as I'm sure you do probably even more frequently than we do, is, what did this bird die from? Or they send you a trail camera photo. What does this bird have?
Yeah.
When they're exhibiting those external symptoms, is it correct to assume that that a lot of those cases are probably avian pox? Is that the one that tends to be have the most commonly physical manifestation that people would pick up on?
Yeah, so pox virus is definitely going to be the number one thing people pick up on just because it's so easily seen
Hmm.
on the non-feathered regions of the bird,
Right.
and can be quite prolific and quite obvious. Having said that, I've had birds submitted um, that grossly looked identical to avian pox and turned out to be something else.
Hmm.
But for most part, that's what my initial differential would be is avian pox virus. Whereas some people might find a turkey that's found dead, skin looks perfectly normal. They may just assume, death happens, no big deal. Well, internally, there could be something really important there. So I always suggest that whenever anyone sees a sick or dead animal that's not too rotten, to try and get it to a diagnostic lab. Or if you're in the field and you have a field necropsy kit, which includes some formalin, which is used to stop the decomposition. Um, you can take some samples if you just have to take a few samples. And the great thing about most of us, we all have cameras now on our cell phones,
Hmm.
so you can take photos uh, and send that to me to look at grossly, and then I can look at it under the microscope, the samples you take in formalin, and we can match that up. So, um, or worse, preferably, I would rather not the carcass be frozen because it can make it challenging. But if I'd rather be frozen than not look at it at all
Hmm.
and then submit it to us. So yeah, whenever there's a chance, and the more we get of those cases, it might only be a one-off here and a one-off there, but as time goes on, we build that record, patterns emerge, and that allows us to really see over time, over certain geographical areas, certain age, certain sex, whether or not we're having some patterns emerge as we get more of those cases come in.
Mm-hmm.
So, Rick, I get plenty of those kinds of requests, and I don't know what to tell people often when they send me a trail cam photo because what you're describing. But the other question that I get often, and I've heard a few disease ecologists say something along the lines of "we don't know," is they're wondering where did LPDV come from and how is it being transmitted. It sounds like there's difficulty culturing it in the lab. So what do we know about LPDV in terms of where it came from and how it's spreading to infect that much of populations?
Not much, unfortunately, which is frustrating. We don't know where it came from. We, recognized it, 30 some years ago, 30, 40 years ago. And like I said, it's very pervasive throughout the environment and turkeys. You can, as you sample them, you're going to find that in perfectly normal hunter harvest turkeys, you're going to have upwards of 90% of them positive. Transmission, it's hard to know whether or not it could be potentially biting insects or potentially even ticks or other things, whether or not there's some evidence of what's called vertical transmission, where potentially the virus gets inside the egg and the female's oviduct, and maybe they're actually born with it. Hatch with it. And so, we just don't know. It's one of the big frustrating causes. I feel like when I get asked LPVD that I could throw a dart at an answer board and it'd be as equally likely to be true as whatever I can say, because there's just so little information out there on it.
So, I think that's remarkable to me and probably a lot of our audience that we have a whole bunch of people like yourself that work on disease ecology, and to not have better information on that is interesting. I guess are there some leading hypotheses, or can you explain to us what is the difficulty in to getting that information?
Yeah, I would say the difficulty is, so whenever you have a disease that is extremely common in a population, it's very easy to find correlations that are not causations. We see that in certain other diseases. One I work on a lot is toxoplasma, which is a protozoal parasite, be very common in certain species. And it's easy when you get that common, particularly without clinical disease, to try and find all these correlations that it's impacting, it's associated with lower recruitment, potentially less numbers of turkeys. But that's a stretch, and it's not scientifically sound to try and make that connection there. And so that's been one of the biggest challenges is trying to understand, wrap our minds around how can we have such a high level of turkeys infected with this, so many of them that have absolutely nothing wrong with them, whether you're looking at them with your gross eyes or under a microscope or doing other testing. And then what is it doing to the population status? Is it just a virus that is there and there has to be a secondary stressor that we don't know about that happens very rarely that leads to the animals having clinical disease? We don't know. And so when we can get over the hurdle of figuring out how to grow this virus in the lab so that we can do experimental infections with animals, we will really be able to answer these questions. And that's just time. Unfortunately, there's really no way to get around that except trial and error. And trial and error takes funding. And people willing to say, I'm willing to have this experiment fail, not to fail as in give up, but the feedback so that the next time I try something different until I figure it out. And that's how science works. And so it has to be that mindset to be able to make the testing and the understanding of the disease. As far as finding out about where it came from, we can do some molecular epidemiology. And what I mean by that is we can look at the virus, we can get the DNA sequence of it, and we can look at how it changes over time and changes from different regions. And with that work, we may be able to come up with information regarding where it likely came from, or at least, what are the major transmission routes and where do we see the same sequence or strain in a certain area, or is it multiple strains happening? So I know that's something that the University of Georgia folks, Nicole and other people, are working on right now.
Yeah. Rick, one of the questions that I get sometimes about, you know, testing for, prevalence and geographic distribution of disease, um, is, folks will oftentimes say, well, you probably aren't finding a lot of positives for this or that because those birds, birds that are infected with that are just not going to show up in your sample because they're out there dead and they've probably been scavenged, right?
Yes.
So, do you think that maybe some of that is occurring with LPDV as well? Like, could it be that not necessarily that those birds aren't available for the sample because we obviously see really high prevalence rates in the birds that we test that look otherwise healthy, but is it just maybe that the really sick ones aren't showing up for some reason?
That's possible. I guess where I may the only thing that we could use right now to tell that is the birds that we have some sort of um, telemetry or GPS,
Hmm.
that we can actually get a mortality signal,
Yeah,
and be able to actually get those samples to a lab. I think that happens for a, a, a good number of diseases. I don't have any evidence or thoughts to say it is happening for LPVD. Um, there probably are birds that are dying of it that are sick that we
sure.
just never know about it. Um, I think of another disease that's probably even more likely to have that situation occur, where birds die before anyone ever finds them. And that's the blackhead disease, because
Yeah,
it's such a rapid disease. From the time that they show clinical signs to death can be hours. And so I imagine a lot of those are they're scavenged before you ever know they had it. So
yeah.
um,
So we, based on your recommendation, we tested several hundred birds here in Alabama for blackhead, and we only got 3 positives.
hmm,
And all those birds had outward symptoms.
yeah. So you actually got kind of lucky. You caught them at a very short window of time.
Yeah.
We actually did experimental infection of domestic turkeys, which I know are different, over in our research facility, looking for a drug study. And my grad students had never worked on blackhead before, and I had told them, you're going to go in one night and they're going to be healthy. You can go the next morning, they'll be dead.
Wow.
And they didn't believe me. And sure enough,
Wow.
I got a text one morning like, "Oh my God, there's like 12 dead birds today." And they all had blackhead. The night before, they were fine. Um, the only thing you might end up seeing a little bit of, a little bit of sulfur yellow diarrhea. That's one of the signs they get a blackhead, because the protozoa parasite infects the liver, and impedes the ability of the liver to break down the red blood cells into its normal components. And because of that, that feces, instead of having that brown, the black color, has that kind of sulfur yellow. So that's very characteristic. But yeah, it's incredibly rapid. We had several samples from our study turn up positive. One was a poult just acting sick, obviously just really lethargic, about very moribund, in the middle of a road, and the grad student was able to get it, euthanize it. And then we had a few other ones that were found by landowners just dead,
Mm-hmm.
and has turned out to be blackhead. It could have been, I'm sure, other diseases as well, but I think that one's just so surprising how rapid it is.
Yeah. Yeah, for sure. Um, Circling back to LP, I like that you call it LPVD, by the way, because
Like,
disease virus never has made sense to me. So viral disease makes way more.
yeah,
But
I've heard people saying it both ways, and I thought it was like just my dyslexia that I couldn't
yeah,
keep up with which order they were.
disease
But go ahead.
virus just doesn't
yeah.
roll off the tongue. But in domestic birds, what are their outcomes like? Because that it's I mean, it's been known to affect domestic poultry flocks for a while. Is that
Yeah.
correct?
Yeah, so it's kind of similar to them too, that you can get, those with the tumor-like lesions in some of their organs. You can get some of the bone lesions, but you also can get birds that are perfectly normal. So
Mm-hmm.
it seems to kind of follow that same rhythm and rhyme um, scenario of having certain ones that get disease and certain ones that don't, and
Gotcha.
it's definitely a mystery. Yeah, I should. It's one of those things where LPVD is going to could be someone's career, and it could also make them retire early. So because it's good, the more you know, the more frustrating it can be. But we do need to know more about it, and I'm confident that we are going to figure a way out to do some more ways. What the Nicole and her group have done is they have taken some of the tumor-like material from birds that had clinical symptoms, and they grind it up, made like a emulsification of it, and use that to feed other birds. I've also done it where they kind of made a scar on their skin and gave it to them. That seemed like that caused the more lesions. So that's maybe why I thought it might be more associated with biting flies.
Mm.
Um, but some of them did come down with clinical symptoms when they did that. However, we don't know if that's real or not. Is that transmission how it occurs in the wild? It's
Right.
completely unknown.
Well,
So
Rick, is it possible that the subset of individuals that have clinical signs is because of the way they got it?
it potentially, yeah, it could be that. It could be the host genetics. Potentially, they have what's their different the genes that encode their different immune system
Mm-hmm.
proteins are a little bit
Yeah.
different. It could be the way they got it. It could have been concurrent issues. It could have been nutritional status. And it probably is a combination of all those, to be honest with you. It's
Mm-hmm.
probably
Yeah.
a multi-combination thing. That's usually
Yeah.
what we find when we do stuff like this.
Well, it seems like things like this that are particularly daunting, like you can't figure it out, it's because there's so many factors going into it. You can't isolate one as being most important because it has
Yeah.
to be an interaction.
Yeah, no, it is. It's multifactorial, which is also is challenging to understand the full ecology of the disease.
Mhm.
So
Well, kind of going back to something else that you talked about that I think is potentially relevant here, when you were talking about aflatoxin, a lot of the effects of aflatoxins are just, I don't know, subclinical, I guess, where you can't visually see it, but makes the bird more vulnerable to other things.
mhm.
I mean, could that be at play here?
That's an excellent question, and that's one of the things I had dinner with Nicole at the NWTF convention last month, and we were talking about our collaborative research that we can do once they can get their infection model LPVD, and that would be having concurrent infections occurring there,
Hmm.
or giving them LPD first and then giving them something that generally is not considered too pathogenic, and seeing if the immune system just so weakened that they actually get disease from it.
Yeah.
Um, and so, yeah, I think that's an excellent point. Is what exactly is the whole system there?
Hmm. Well, regarding the aflatoxins, I had another question about that. I know there's been some work where experimental work like what you're talking about, where they fed turkeys different levels of aflatoxin and then looked at the outcome. And I know that the levels that they fed them in that in that study are commensurate with the levels that we commonly find like in a feed pile you talked about earlier. Is that, I guess, could you talk about that? What are the strengths of those? What's the strength of the inference you can make from that, and what are the fallacies? Because
Yeah. So I really like those studies where they have experimentally and they can actually test and know that this level of corn has this high of alpha toxin, and then maybe actually even getting down to even other mycotoxins that could be there. Um, and then trying to figure out what does that mean in the wild. So, in a captive situation, particularly wild turkeys, they're so stressed out inside a lab that their immune system is already going to be, having an impact because of that. Um, However, it does give you at least some guidelines of saying, here's a level that we don't see anything. Here's a level where we start seeing a little bit of liver um, lesions on the microscope. And here's an area where we can look at it and see the liver looks horrible. And then we can potentially have some other components there. So I think a lot of those things have to be a combination of lab work that has to then try to be proven in the field.
Mhm.
So I think just having work in the lab without field data to back it up um, can be a red herring to some degree. But I also think field work by itself without the lab work can equally be a red herring. So I think kind of simultaneously in parallel, looking at, samples in the field, testing corn that may be placed out for birds, um, getting aflatoxin levels, and, particularly if you have GPS trackers on these birds, that you can have mortality signals, and then lining that up to what you see in the lab over time, that makes a really solid argument. Um, and it comes down to one of the biggest concerns I have in general wildlife disease ecology is just the concern of artificial feeding of wildlife. Period.
Hmm.
I know it's seen sometimes as a benefit because you get to see the animals, you might have what the human wildlife bond. But whenever you start bringing animals into higher densities than they normally would be in, you really increase the transmission of multiple pathogens, whether it be fungal, bacteria, viral, parasitic. Um, no different than having a really large group of daycare kids and you send one kid to school that day that's got the snotty nose.
That's a particularly poignant example for me and
Yeah,
Marcus
I
right
know,
now.
Yeah.
I know.
Well,
Um,
Rick as well. I mean, it
so
seems like every time we turn around, we have whatever the global pandemic is in the
Yeah.
school, and we are at
exactly.
home,
So
home again.
you're doing the exact same thing with these animals that are not meant to be in these, high density areas, whether it be feeding deer, turkeys, even songbirds. We're actually finding that you know, feeding of songbirds, if you're not keeping your feeders clean, it's
Mhm.
actually contributing to some of the most important diseases right now. Uh, Probably nowhere near as important as feral cats, outdoor cats, but at the same time, it's not trivial.
Hmm.
You can have salmonella, uh, aflatoxins, trichomonas, all those things be really prevalent in water baths and water feeders that are not kept clean.
Mhm.
Um, and so it's just again just bringing a lot of animals into a one location. So I really caution people about artificial feeding, um, and I often really encourage them as we should is let's improve the natural habitat for them
Hmm.
and
Mhm.
let's work on that. That's the long term gain. Your short term gain that probably cost you more in the end is your artificial feeding.
Have you?
We certainly have
Yeah,
covered that in the
we've
Yeah,
been
same
banging that
vein.
drum for a while.
uh,
Yeah.
most people have. So
Have you, Rick, seen any birds, in the lab that have come in, wild birds that you were concerned that aflatoxins may have been the cause of mortality? Or is that just
not
too
in,
hard to differentiate from everything else they have going on?
yeah, not in my time. I haven't. There was a case that my supervisor, Randy Davidson, he used to be one of the professors at Squidus. There's apparently a case. I can't remember exactly when it happened, but a railroad car of corn had overturned somewhere not far from Athens, Georgia, and the corn just lay there and got moldy, and quite a few turkeys from that region had died of aflatoxicosis. They were brought in
Mm,
from the lab. But myself, I haven't. And again, it's probably not the overt issues I worry about aflatoxin.
right,
It's those subtle things, whether it reduces potential fertility or has an implication in reproductive hormones or all
right.
those things that those fine tune issues we have to have our population growing.
Yeah, that makes
Mm.
sense.
Well, I have heard from at least one person who submitted some turkeys to Squidus, and they came back with aflatoxicosis.
Yep, I
And,
actually
th-
have. Go
and
ahead.
those were found dead, Marcus?
Yeah, I
Yeah,
believe so.
Yeah, I wouldn't be surprised. They're out there for sure.
yeah. So, we talked about histomonas briefly just a little while ago, and I definitely wanted to circle back to that one because the chicken litter question is one that comes up a lot
I was
with the
waiting
general.
for you to bring that back up with.
Yeah, yeah. So we get questions about that a lot from the general public. So we wanted to make sure we spent a little bit of time talking with you about it today.
For sure. I'd rather talk about that than LPVD. I feel like I know at least. I
Sorry.
feel like we at least know something about this. So, histomonas is actually a fascinating disease. It's a disease that wasn't believed to be in the North America until pheasants were introduced into North America because they are the natural host. Sort of like how we talked about white-tailed deer not getting sick from brainworm. Pheasants can drink histomonas all day and they're not going to get sick from it, but they can shed it in their feces. Um, and the parasite has evolved this kind of really crazy life cycle where it actually can invade a separate parasite. It can invade a nematode worm of the cecum called Heterachus, and it can get inside the eggs of that nematode. Again, those eggs are microscopic, and they can stay dormant in the environment inside those eggs for a very long time. Now, outside of that egg, histamos is very fragile. It will not survive in the environment very long at all. But inside that heterachus egg, that evolution has occurred that it can stay dormant there for years. And then those eggs, or the heterachus worms in general, they can be ingested by everything from earthworms to beetles to other arthropods. So then potentially when a bird, whether it be chicken, turkey, pheasant, quail, whatever, ingest one of these arthropods, inside the arthropod is the heterachus egg, heterachus worm, and inside that's histomonas. So we often refer to it as that rushing nesting doll situation. If
Mm,
you've seen those when one doll instead of another instead of another. Um,
That's
and
wild.
so so that's kind of how I have the students remember it, is having that. So we've done some work. We actually have found the DNA of it inside beetles that have come out of poultry houses as well as that. So one of the things I wanted to make sure I talk about is that not all poultry litter is the same, and that's really
hmm
important. Um, the high majority of poultry houses in the country are what's called broiler houses, and so broilers, they have a really short lifespan. By the time the chick goes to the house until it goes to market, is only about 6 weeks. And so it doesn't give it time for the heterachus life cycle really to be completed. It doesn't really, and so they're probably really don't have much implications in transmission. Now, on the flip side of that is a group of that's called the breeders, or sometimes called the broiler breeders. And so these are the birds that are producing the eggs that become the broiler chicks. These tend to be long-lived, potentially up to a year. They have the replacement pullets, and then they eventually grow up and become. And so they're kept on floors, litter floors, and so that transmission, that is sufficient to have that. And we have done some work looking at intestines of some of these broiler breeder chickens that are sent to slaughter after their lifespan, and found they have a decent level of heterachus in there. So a lot of things have changed over the last 15 years or so with that, and it includes everything from the drug that was initially used to control heterachus, fenbendazole. It seems that there has been growing selection of resistant strains of heterachus. So the drug doesn't work nearly as well, even in some cases a 2-time dose. The arthropods that, particularly darkling beetles and other arthropods that are inside poultry houses, the insecticides they have used, again, their resistant strains of those beetles have been selected. And so they have a very high level of beetles in there. And then changing the litter has the duration of that has increased, where they often do top dressing. And then when these houses are cleaned out, there's no regulation of them having to compost that litter before it's used for fertilizer. And so, as the price of conventional fertilizers increased, more and more people are using chicken litter as a source of fertilization. And so the concern, obviously, is whether or not these wild turkeys, could be ingesting something in the litter. We did some experiments here at our university here, where we took a commercially raised version. It's a heritage breed that looked like a wild turkey, but obviously not a wild turkey. That's the closest I could get. And we placed a subset of them on spent chicken litter that we got from a breeder house in Middle Tennessee. And then we had our control birds that were placed on clean shavings. One of the birds on the litter, the spent litter, died of blackhead about day 13 in the experiment. All birds were euthanized day 15. Found about 5 birds in total, including the one that died, were PCR positive for histomonas. And we also took a subset of those beetles that we found in that litter and did PCR. About 5% of the darkling beetles were PCR positive for histomonas. So it really did show a connection there. Am I ready to say that is the cause of turkey decline? Absolutely not. I don't. I think it's something that definitely warrants more attention and warrants more concern. And it's something that we looked at one house at one time point, and you really can't make too much emphasis. But again, it leaves me enough to say it definitely deserves more attention, more research. And unfortunately, it's been challenging to do much more there. The funding that we got from the Tennessee Wildlife Resource Agency has expired. It was a wonderful grant they got us. And so it's something that has to be done. The other question that we don't know about histomonas is, are all strains of histomonas equally deadly to wild turkeys? No one knows that. And so what we need to do there, and something I've been wanting to do,
I didn't even know there were strains.
Oh yeah, so it's actually quite genetically diverse. I worked on a paper as a PhD student with a master's student. We looked at genetic diversity of histomonas. It's quite diverse. And I have strains of the parasite. This one we can grow, frozen liquid nitrogen in my lab. And what I wanted to do is collect wild turkey eggs, hatch them, and then allow these poults to grow up to maybe about a month of age, and then infect certain number of poults with the different strains and say, okay, well, yeah, all strains cause mortality, and then say, no, only this one caused mortality, the rest of them didn't, because we have no idea if it's uniformly. And the reason I say that for is I have gotten hunter-killed birds, adult male hunter-killed birds, obviously male, um, that we were, the hunter took the breast, we were given the rest of the carcass. I took the part of the intestines called the cecum, it's a blunt-ended pouch, and that's where histomonas first sets up shop when it gets infected. And we were able to get genetic material that was indicating that bird had histomonas in its cecum. So the question was, this was a hunter-killed bird that came to a hunter that was calling. Does that mean this bird had histomonas and wasn't having disease? Or was this bird just at the very early stages of histomonas? And if hunting season was 2 weeks later, would that bird have been dead of blackhead and not been harvested? And I cannot answer that question because we have never done the experiments to do that. So that's a really important experiment that needs to be done. But that takes funding and takes time, time to collect the wild turkey. And I want to do this in real wild turkeys. I want to be able to collect wild turkey eggs, hatch them, and do this work. So there's a lot of interest in it. I'm not gonna, like I said, most chicken litter is broiler, so you're safe with that. Broiler breeder, I would just have to ask where the source of the litter comes from to know the potential implications of it. And the other thing is, I'm working with a microbiologist here, environmental microbiologist, because we recognize the importance of using this as a fertilizer. What if we can figure out the parameters of composting that inactivates the heterachus and histomonas that we can actually incorporate? And so we're waiting to hear from USDA if we got funded for a grant to look at that, because that would not only help wild turkeys, that would also help domestic birds as well.
So are you talking about like looking at best management practices for composting to ensure that
exactly,
it's not infectious?
Exactly, ensure it's not infectious. And this is what he does for his career. He has sentinel chambers, and he has different ways to measure moisture, heat. And he's done this for a lot of different diseases. And so we're hopefully get funded for this to do this work.
Yeah, I think that would be really good information for managers to have. I mean, even if they only had access to, like the broiler breeder house litter and they wanted to fertilize with it, maybe they just get the load, dropped off under a pole barn or something like that, and they compost it for a period of time before spreading it just to make sure.
Absolutely. And that's what we want to be able to do is kind of have a cheap way to know that if you have it at this moisture, at this temperature, you're going to be able to inactivate it. So, yeah, we've been looking for quite a few people to try and fund that study as well. It definitely needs that one. I think is a slam dunk. That's a win-win. Because the one thing I didn't say is, historically, chickens were considered similar to pheasants, refractory to histomonas. That has changed. In the broiler breeder industry, the replacement poults, blackhead is now the number one disease of replacement poults. Something has changed, whether it's genetic drift of the birds or something else, that's actually causing quite a bit of disease in the replacement pullets, not poults, replacement pullets, that is actually causing a financial impact. So it could be helping the producers for the breeder industry equally as much, if not more, than conservation of wild turkeys, for whatever impact blackhead may have on populations.
Yeah. Well, I hate that that's happening to producers, but at the same time, I guess the silver lining is that money will be going towards that.
We're hoping to. We're hoping that it's seen that this is something that we'll be going to. So, so we'll keep our fingers crossed that we can get some of these grants funded here in the near future. And another work I'm doing with that might be interesting for some of the viewers is I I've actually very fortunately became introduced to a new assistant professor here, and her line of study is forensic entomology. She looks how different insects change on human corpses over time that can be used to infer how long something's been dead or has it been moved or what has gone on. And so, what we're actually hoping to do, her and I working together, is maybe start in certain areas where we're having population decline, is put out very inexpensive traps for blowflies. Because if you probably known, something dies, blowflies are on it before the thing takes its last breath.
Mhm.
And see if we can grind those blowflies up and find the DNA of what it ate, potentially the pathogens. And that's not only a really important for potentially histomonas, but that could be a game changer for almost all wild diseases that have DNA, which pretty much is everything
Hmm.
except prions, DNA and RNA. Um, and so you potentially may be able to go somewhere and catch X number of blowflies, do molecular testing, and say these are the diseases circulating in this group of animals right now. So,
So
so
would you,
anyhow,
with that, would you be able to say, okay, the blowfly was eating a turkey carcass, and the turkey carcass had these pathogens associated with it? Is that what
yeah,
you're saying?
yeah, because you can do molecular testing to confirm turkey DNA. First of all, know what it ate, and
Yeah.
then you can do a separate molecular test for whatever. You could be looking for LPVD. You could be looking for histomonas, pox. Um, Toxins wouldn't work. I don't think they would work. I mean, maybe unless they accumulate in the fly itself. But pretty much anything that has any infectious disease other than a prion would be able to be detected. And maybe someone who knows more about prions could even do some Western blotting or something on it that would be able to show.
That would be a lot easier to go about than collecting the 900 carcasses that we did over the past couple of years.
Yeah,
Just
yeah.
trap some flies.
I know.
Just go feed a
We're
put a chunk
excited.
of turkey.
Everyone I send the proposal look at, my department head around also goes fascinating and very concerning. So but
Things
concerning
are getting weird now.
in
Yeah,
that you're like we're going in this realm of but I think it gets to be a huge game changer for us.
yeah,
So
for
I'm
sure.
excited to see what it brings.
Hmm. That is pretty wild. Well, I don't know how much time you guys have, but I also would like to talk about the egg study that
Yeah,
you have going
I'm
on.
good to go.
yeah, I've got
So
a few more minutes. We can talk about that. I definitely want
yeah.
to hear what's going on with that.
Yeah. So so one of the things coming out of this project we've been working on with TWRA for the last 7 years now is understanding some of the recruitment issues. We definitely know from the work that Dr. Dave Bueller and Craig Harper have been doing that we're having poor poult recruitment, we're having poor nest success. A lot of all the benchmarks that we need to hit to have a stable population, we're not there. Um, but one of my questions has been, and it kind of also revolves a little bit about this 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 discussion now about are we potentially killing too many dominant males too early in the season that we unfortunately are not allowing uh, ample coverage of the hens to produce fertile eggs. And so there's been all these areas of discussion come around. And, we thought about, how can we do this? Potentially we can go candle eggs at a nest. That's be really challenging. And I also don't want to bump a hen off a nest. And so in the midst of this, the PhD student I have working on this project, Laura Horton, um, she likes to spend her late evenings, early mornings searching the web for science, and she came across this really interesting site at the London Zoo where they're using this technology to understand fertility issue in captive bred birds that are threatened and endangered. Uh, because I had a decent number of eggs that wouldn't hatch. And the long standing theory was, well, they're infertile because I can crack it open and all I see is a fertilization disc. And if it was fertilized, there should have been actually an embryo there. Well, anyhow, those researchers there said, well, let's actually put some science to it. And so what they did is they were able to remove the fertilization disc from the yolk and place a DNA stain on it. And then under the fluorescent microscope, the DNA stain is uptaken by either sperm or uptaken by cells of the fertilization disc that may be starting to divide. And so suddenly, what you can answer is two questions. One, was that egg fertilized? Is there evidence that there's actually sperm trapped in what we call the vitelline membranes, which is the membranes that cover, you have the inner vitelline membrane, and then you have sperm is uses to fertilize, and the outer vitelline membrane comes along after fertilization. And so you can visualize everything, the holes made in the vitelline membrane by the sperm, but also the trapped sperm. And then you can look at the fertilization disc and say, was there evidence that there was starting to be cell division and it stopped? Well, sure enough, the people in the zoo found out the vast majority of the eggs were indeed fertilized, and there was initial embryonic division, and there was, for some reason, there was early embryonic death. Exactly why, they don't know that. And so I said, you know what, we got to try this. We got to see if this works for turkeys. So I actually got some of my colleagues at the poultry science department sent me some fertilized chicken eggs. I had some fertilized duck eggs at home, and I also have a single female hen domestic turkey at home, so I know she's not. And so I got eggs of different ones, and sure enough, the chicken and the turkey duck eggs, you could see the sperm, so much of it, and you could see where they started having some growth. And obviously, the turkey, since she wasn't fertilized, there was none. And it has worked so well. So this past year, we did our first trial funded by the state chapter of the NWTF, um, looking at a few. I think we looked at about 8 different states, and found majority of eggs were fertilized. Now, most of these eggs came from nests that were either abandoned or, unfortunately, a farmer mowed the field when they probably shouldn't have been and killed a hen. Um, and so we recognized they were fertile. What we're really interested in is getting those eggs where you come across, and it's got to be just get lucky. I know that, particularly with telemetry or not telemetry, I guess, where you have a subset of the chicks that hatched, but some that didn't.
Hmm.
To know those ones that didn't, the hen was there long enough to incubate them. Why didn't they not hatch? Were they not fertilized? And so we can look at those. And so this we got funded now for 3 continuous years, starting this year and 2 more years after this, by the national NWTF to look at the study, looking everything from fertility rates. Also, if we have evidence that it was fertilized and the eggs were adequately incubated, and we can see early embryonic death, we can start tracking down different avenues, whether it be looking at aflatoxins in the eggs. We're working with Iowa State to see if their toxicology lab can validate a study. We can potentially do some PCR work on the egg to look for pathogens. Um, but then we can compare that to everything from looking at individual states, as when does hunting season start compared to breeding season? What are the bag limits? Um, what are some of the other factors that may contribute to that, to see, is there really an issue with us killing these dominant males? Um, is that really a problem? So maybe states that have a relatively earlier hunting season than others compared to the breeding season, start of the breeding, is that a concern? And should we be delaying the hunting season? Um, or do we find that's really not an issue at all? And is there any particular reason that we could be having early embryonic death? So it's really cool. It's really kind of cutting edge. It's a great work for undergrads. They get to do, it's not technically that hard, and they get to see things on the microscope and get an answer right away. so rarely in science do we get, instantaneous gratification, but this is one of the few things that we do. So it's been exciting. Laura did most of the work last year as part of her PhD dissertation. She's graduating this summer, so hopefully finding someone else to pick this up. Have one technician on. She's going to be swimming in eggs here in a few months. But luckily we got a good team of undergrads that are excited to work on this. So that's the general gist. So I can tell you a lot more in a few more years after we be able to look at this and analyze it over a couple year period, what it really means as far as fertility rates and any particular thing that may be limiting um, uh, embryo growth.
Mm-hmm. That's going to be super interesting, especially how you're pulling in those pathogens and toxins into the work.
Yeah.
So it's kind of a
Yeah.
comprehensive perspective. That's going to be fascinating.
Yeah, Rick, I really think that's important given, one of the prevailing hypotheses right now is related to hunting and the timing of hunting.
Yeah.
And I think that's really important. I'm excited to see, what comes of it. I know last year we gave you some eggs from Florida, and I know that you said, what, like 8 states you got
Yeah.
eggs from. I also know that a lot of people doing turkey research are listening, the state coordinators in many states. And do you need eggs from people? Like,
Yeah.
how are you going about that?
Yeah. So anyone interested in this project, I would say have their state turkey biologist contact me. You can also contact Roger Shields, who's the state turkey biologist for Tennessee. Him and I, or Derek Keir, uh, or and any of us will get the information. What we have. We have a card on how to label the eggs, instructions on how to preserve them, either short term in the fridge or long term in formalin, to get it to us. Um, let's see what else. We also exactly what information we need from them so that we can be able to understand how it relates to turkey hunting regulations as well as location, stuff like that. So yeah, have them contact us. Particularly contact us early if there's any kind of permits we need from a particular state for these eggs. We can work. Sometimes the university wants a contract in place for that. But otherwise, yeah, we'll take them whether they're individual farmer finding them or a biologist finding them with a telemetry study, or someone just happens to walk across a turkey nest in which there's a few eggs that didn't hatch. Um, all and any of that is valuable information. Every single individual data point is going to make it more and more robust.
Yeah, and if people contact us about it, we'll be sure to forward them to you.
Yeah, please do.
So I'm excited about that.
Yeah, for sure. Would you
Important
want
question.
to provide any kind of contact information if folks are interested in reaching out?
Yeah, absolutely. Probably easiest thing is my email. So my email, is there something to put in a chat or anything I can do?
We can put it in the show
Okay.
notes.
Yeah. If you
So
provide it to us.
yeah, it's just R and then my last name Gerhold, GERHOLD at utk.edu. So the UTK is University of Tennessee at Knoxville. Just wondering what the K meant. .edu, and yeah, you'll get me. So
Perfect.
Yeah, great stuff. We'll have Charlotte put it in the show notes so
Okay.
that anybody that wants to find you,
yeah, please
they'll have
do.
that.
Yeah,
Yeah.
happy to talk about it. I will tell people that I'm currently trying to get 5 PhD students graduated in the next 3 months, 3 to 4 months. So I'll do my best to get back to people.
Good luck with
Yeah. So, but don't take it personal if you don't get a quick response from me.
Yeah, I can certainly appreciate that. But hopefully, they will all be successful, and then you can bring in your new cohort to
Yeah.
focus
No,
on turkeys and all the problems with turkeys.
I luckily have the 5 students I have are all equally amazing, and I'm actually sad. I'm happy for them, kind of sad for myself because I really enjoyed mentoring them the last 5 years. I don't know if it'll ever happen again. But yeah, we've got people working on everything from turkeys to brainworm to coyote diseases. Trying to keep up with them is my biggest challenge because they're so productive. So,
That's awesome.
yeah.
Well, this conversation was long overdue, and it didn't disappoint. We're really glad that we finally were able to connect and have you on, and I learned a lot today, and I'm sure our listeners did as well.
Excellent.
Yeah,
Well,
for
it's
sure.
my pleasure, and I'm happy to future conversations come on again.
Yeah, we are definitely going to want to keep up with what you have going on because you're answering
definitely,
some
and hopefully in the
important
next
stuff.
several years we can really understand a little bit more between LPVD and blackhead and some of these other things, and kind of get a clearer picture of what's going on. So it's kind of nice to see this work in turkeys, and it's definitely needed because there's a lot of concern about
Mhm.
populations right now, and there's so important in the economic, but also just the social of family spending time hunting and the importance of hunting. And I'll share this quick story before I go, but
Please do.
I the very first year we were doing our hunting check station in Middle Tennessee, we set up a shop with the TWRA outside this little gas station where we had advertised, and the hunters brought birds. So there was this flock of people, no pun intended, flock of people that came in to see what we were doing. And this one guy came to me and he's like, "Are you going to figure out why our turkeys aren't doing as well?" And I said, "Well, I'm going to at least do my best to understand the general disease, health issues, as best as I can." And he's like, "Well, I really hope so, because I swear to God, if I can't turkey hunt, I'm going to probably go kill myself." And I was like, "You can't put that pressure on me." I was and I was like, I recognize it's so important. And he's just like, "It's all I want. It's all I live for." And I was like, "Well, you might try fly fishing. It's pretty cool."
Agreed. I love fly fishing also.
Or you might even take up sewing like Marcus.
Yeah,
apparently
exactly.
No,
It
this is fly time, guys. Come on.
It just shows how
I see all that
passionate.
thread in the background.
Yeah.
It just
Yeah.
Underscores what you guys already know, how passionate turkey hunters are.
Yeah. It's very important to many people for a variety of reasons.
Yeah.
It's really been special to me, being part of this podcast and seeing the response, like the passionate group and how supportive they've been of all this research going on. By and large, people really care and they're behind us and trying to help any way they can. So, it's inspiring in some ways.
Yeah,
just to see how passionate people are about the resource.
For sure. Well, this has been wonderful, and let me know in the future what I can do. But I'm excited. Don't think I want to hear myself talk, though. I just feel like that's a dumb thing to say when I hear myself talk. But I enjoyed it very much.
Yeah, we did too. And if you ever come across anything just really interesting that you can't wait to share, make sure to hit us up. We'd be glad to have you back on. So
sure, absolutely.
And I'm sure we'll be, trying to check in regularly because I'm really interested in what you're doing.
Well, thank you, everyone. Thank you for having me on, and it's been a wonderful experience. I'm looking forward to the future podcast with you guys.
Yeah, we appreciate it, Rick. Thanks for coming on.
Of course.
Thanks, everybody.
Wild Turkey Science is part of the Natural Resources University podcast network and is made possible by Turkeys for Tomorrow, a grassroots organization dedicated to the wild turkey. To learn more about TFT, check out turkeysfortomorrow.org.